pleasure for a number of years now. I'm close to graduating, which is this is a very wonderful opportunity for that reason as well. It feels like an accumulation of my work. Why zebrafish? Maybe some of you here are still considering starting to work with zebrafish or are new to zebrafish, so I'll run through this very quickly. They're a very social species, which is my area of interest. They have a high genetic homology to humans, and one fun thing is you can easily administer water-soluble drugs to them. Our lab tries to understand behavior by, tries to understand the function of the brain by studying behavior. So the way that I started my studies is by trying to answer the question, how do zebrafish begin to shoal? Larval zebrafish were previously observed to not really show any grouping behavior, whereas adult zebrafish do. And it was our mutual interest, Dr. Verlei's and mine, to try to investigate how that actually happens, what happens in that period between the larval stage and adulthood. Now what we found is that there was actually a pretty gradual transition from, let's call it no shoaling to shoaling behavior. In this graph, excellent, I don't have a laser pointer, but this works. On the y-axis, we have the average distance between all fish pairs, and on the x-axis, we have the age in days post-fertilization. So we can see here that the average distance decreases over time. And we see a particular fast development. So we see a particular fast period of development right here from about 26 to 45, 47 days post-fertilization. The picture here is the setup of my experiment. This is an open field, and it shows 10 fish roaming around the open field freely. This is a similar picture, but shows larvae, which unfortunately are practically invisible on screen. To quantify this behavior, we use a fairly simplistic tracking program, which basically just provides XY coordinates for each fish in the tank. Now, the raw data looks roughly like this, and even though you can't read it, you probably wouldn't really make a lot of sense of it if you could read it. It's a bunch of numbers on the screen. Our tracking program is, like I said, pretty simplistic. It provides text output of all the XY coordinates, and it does so for, we get about 13,000 data points per trial, which is about 8 minutes long, because it samples roughly 30 times per second. So you may imagine that with a rather large study, we need some further data prep and analysis. The tool that I used for that is R, which is a programming language, in case any of you is not familiar with it. I will very briefly explain what R does. It's basically a Swiss Army knife for data. It's a great tool for slicing up data, aggregating data, or connecting to data sources. It is also used for modeling computation, statistics, as well as graphing. Now, this is an example of my code, and I'm a little bit disappointed with how it's showing up on screen. I do apologize. Basically, this shows the program that I have written in this case, and this shows the data output. So it's just an example of slicing and dicing the data to put it together into a database that we can actually use to make sense of it. Now, let's dive a little bit deeper into zebrafish social behavior. I use zebrafish as a model for fetal alcohol spectrum disorder. For any of you who are not very familiar with that, this is what happens to children when the mother consumed alcohol during pregnancy. Or, sorry, it may happen to children. It is very unclear what the mechanisms are underlying it, and although it dates back to biblical times. Fetal alcohol syndrome was first described and officially published in the 70s, and ever since there's been a surge in research that is being done on that. There is a range of outcomes that are rather broad, but it is also the most commonly, most common cause for mental retardation today. Its most profound effects are behavioral and cognitive, and it's estimated about 25% of juvenile incarcerated individuals are suffering from some form of FASD. So it's a relatively relevant disorder to study. Now, how do we model this in zebrafish? It's actually surprisingly simple because alcohol is water soluble. We can put alcohol into the water with the zebrafish eggs. It will be absorbed by the chorion, and we can look at the long-term social effects. I've been very interested in looking at very low doses of alcohol exposure. There are a lot of rodent studies and some very few zebrafish studies that have looked at very high doses of alcohol. My interests would range more towards what would happen with moderate to low consumption, which is still socially acceptable in many cultures for pregnant women. So our dosing schedule was a one-time exposure of 0 to 1%, 5 different concentrations. And colleagues of mine in the lab have established that only about 1/30th of that concentration is present inside the egg. So you can imagine it's very low doses. Now, I've previously described the ontogeny of shoaling behavior. That was the graph I just showed you. So we can use that as a bit of a benchmark. This is a trace of one of my experiments, and this is just to illustrate what I can do by using R. By having only XY coordinate data, it's both an advantage and a disadvantage. It's a disadvantage from the perspective that you have to figure everything out yourself, and you don't have a really nice output of your graphs or your data right away. But at the same time, I can set these boundaries to see how much time they spend in corners, how much time they spend in perimeters. We can decide we want to use the outside 10% or the outside 30% perimeter, and I can look at, for example, distance from the wall. So this is, again, just an example showing what you need, the work you need to do in a command line driven system to figure out the how much time it is the fish is spending in the outside 10% perimeter. And this shows the distance from the wall. Now, for some actual results, because that's obviously what everybody hopefully is interested in. On the x-axis, again, we have time. This is age in days post-fertilization, and to fit everything onto the screen, all the numbers are rather small. But this starts at 7 days to 91 days post-fertilization. On the y-axis, we have the average distance between all the fish pairs again, so a measure of shoal longitude. In the beginning, when we are looking at the larvae from 7 days to about 20-some days, we don't really see a lot of difference among all the treatment groups. The white bars is the control group, and all the gray scale bars are the treatment groups ranging from 0.25 to 1%, with the black being 1%. We start seeing very significant differences between our control groups at 47 days post-fertilization. Yes, at 47 days post-fertilization. We start seeing a difference between our control and lower dose alcohol treatment groups and the 1%, and it only becomes more robust between the controls and the alcohol groups as time progresses. In addition to seeing a reduced amount of shoaling or reduced shoal cohesion in our zebrafish over time, we also see a difference in the average and maximum inter-individual distances. This is a mouthful just to describe the maximum distance that these fish are apart at any point in time during the trial. The green line is the 1% treatment group, and the red line is the control here. So we can see again age on the x-axis, and on the y-axis is the average distance. The control fish, as they age, their maximum distance that they're apart is much less than our treatment groups. I really wish I could fix the color on the screen. I'm very sorry. This is affecting the quality, but I also looked at the distribution of this parameter of the average inter-individual distance. And just very briefly, on the left here, we have a 15-day-old zebrafish, and on the right, we have 90-day-old zebrafish. We don't see a difference in the distribution of the inter-individual distances, but we do see a bit of a slanting on the other side. Again, the green is the 1%, and the red is the control. With the 1% groups, we see more of a normal distribution of this parameter, and with the control groups, we see a skewed distribution. Now, the reason for this is that in regular control zebrafish that are healthy, the majority of them do want to shoal. They're a social species, so they do have a motivation to shoal, which is why we see the skewness. And in the 1%, we see that some of that is lost. This, again, shows the skewness throughout development, and this should show as gray and white bars, and I have no clue why it's not. I'm very sorry. Now, you may argue that perhaps these fetal alcohol exposed zebrafish are just not able to swim in the same way that normal control fish do. So I looked at the speed and at the distance traveled. The top set of graphs is the total distance traveled, and the bottom is the speed of a single fish in an open field, and we do not detect any differences among any of the treatment groups. So we can rule out that as a potential cause for the difference in behavior. Now, looking at the brain, we know that the site of conspecifics is very rewarding, and dopamine has been implicated in the reward system. We also looked at serotonin, which is implicated in fear response, aggression, and other mood disorders. So to look at the zebrafish brain that has been exposed to alcohol, we basically dissected the entire brain, and we used high performance liquid chromatography to look at the amount of dopamine and serotonin present in the brain. And this is, again, done throughout development. At the top left, we have dopamine from the whole brain in 15-day-old zebrafish. Here, 40 days, 70, and 102 days. And what we see, again, is something that, at this point, we can only say correlates to the behavioral output, but it's certainly not a causative correlation as far as we can tell. But at 15 days, we don't see a difference between the treatment groups in terms of the amount of dopamine present. We do start to see a significant difference at 40 days and moving on to 70 and 102 days between the control groups and every other treatment group. So among the different alcohol treatment groups, we don't see a difference. Excuse me. The same story, or at least a very similar story, is present with serotonin. Except here at 40 days, we also don't see a significant difference with serotonin, but we do see a difference in 70 and 102 days, where again, both serotonin and dopamine are depressed throughout in the adult zebrafish and in the young adult zebrafish. So our findings so far suggest that a very, very low dose of alcohol is enough to alter both neurochemistry as well as behavior. Now, I would like to argue that zebrafish are an excellent candidate for high throughput behavioral screening, and as we are moving more into the landscape of video tracking, of automated tracking, zebrafish behavioral assays are becoming increasingly attractive for this purpose. And that concludes my talk. I have to thank my lab members, my committee, particularly my PI, Dr. Robert Gerlai, and the symposium organizers for inviting me, and all of you for being here and hearing me speak.